Welcome to the Synthace DOE Masterclass. Anyone can run DOEs, and I'm going to be your host for today's session. This webinar is the latest on our series on DOE. The main presenter today will be Dr. Nuno Leitão, who is a principal scientist in our labs here at Synthace with extensive experience of DOE and using the Synthace platform. But before he gets going, I wanted to give you a bit of history. So when we first founded Synthace, it was with the conviction that biology could be done better. And more specifically with the conviction that multifactorial methods are dramatically more powerful than standard experimental designs for exploring the interconnected complexities of biological systems. We started off at Synthace with manual methods to run DOEs, but we soon realized that we needed still more complex and powerful designs with the kind of challenges that we're facing with trying to engineer biology. As we attempted these more powerful designs, our time to plan and run DOEs exploded from about 1 week per iteration to over 6 weeks. So where once we were carving our way through design space, we were now getting really bogged down. We were getting really powerful insights, but they were really hard to make happen each time. And this is when we'd moved from running 15-factor experiments all the way up to sort of 27 factors, which remains the most factors that we've been able to do at any one time. And these were designs that we were doing manually. So this is when we started making software, internal software to help run DOEs. And this helped us to do dramatically more powerful DOEs a lot easier. And it's where this Synthace many years later has got to. We're really excited to show you how this software has evolved and got to in this time. And we hope you find it interesting. So now I'm going to hand over to Nuno, who's going to start with a brief recap of what DOE is and why we should all be using it. Over to you, Nuno.
Thank you so much, Marcus. Most of us here are already bought into DOE, but it's always good to refresh why it's such a powerful methodology. So if we think of our classic one factor at a time experimentation, what we tend to do, as the name suggests, is we keep all the factors constant in our experiment and we're varying one. So in this case, if we look here, we are varying factor 1, and maybe in the first experiment, we found that this is the optimal condition for factor 1. Then we need to run a second experiment where we fix factor 1 and we vary factor 2 here in orange, and perhaps we find this is the optimal point. And on the third experiment, we can now keep factor 1 and factor 2 together, and we find that the best value for factor 3 is this green point here. Now, this has taken us 3 experiments to carry out, and obviously this means it's time and resource intensive. But more importantly is that we've actually only explored a very narrow design space. We've completely ignored any possible interactions between factor 1, factor 2, and factor 3. And so there's nothing to tell us that actually we found the best possible solution. This is where DOE can be incredibly powerful. So in DOE, we don't just vary one factor at a time. We explore multiple factors simultaneously, and we use statistics to actually cover a wide range of the design space, still in a low number of runs. This is what we're referring here as traditional DOE. We can optimize multiple factors simultaneously, capture interactions, and have a better understanding of our system. More recently, we started doing what is referred to as space-filling DOEs, where actually we can sample across the entire design space. So we have a much higher resolution of the response that we're measuring. And this is becoming possible these days because we're running in biology assays using automation and miniaturizing. So cost and time no longer are so limiting. But since Marcus mentioned story time, I figured I should share a little bit about the first DOE I ran that was here at Synthace, because as most scientists, I wasn't really educated in DOE throughout in university or during my PhD. And when I came to Synthace, actually, the task that I was given had nothing necessarily to do with DOE. I was asked to implement a robust and precise automated qPCR protocol. Now, by then, all we spoke about was DOEs. I figured, you know what, why can't I bring the two together? I figured qPCR, I had enough experience with qPCR. All you need to do is to make sure that you're pipetting accurately. And that was actually the challenge. So optimizing the liquid classes of the robots. And so I figured I could use DOE to do just that. So DOE for liquid class optimization. So a liquid class usually includes all the factors that control how a robot pipettes. So that's your aspiration speeds or flow rate, your dispense speeds, how deep the tip goes into the liquid, how long you wait there before, after aspirating, before it moves back up. And so the approach that I took was to create a design that would create a variety of different classes. And as my response, I was using the variability across technical replicates in a qPCR plate. So I set up a 384 qPCR plates. All reactions were the same, but every 4 reactions were pipetted using one specific set of liquid classes or one of my DOE runs. And then I was calculating the coefficient of variance between those 4 technical replicates and finding the one that is lowest, because that would suggest that has been the more precise pipetting. This is a summary figure of that work where you can see here across over 80 different liquid classes that were tested. I found one here that has a CV that is so low that you can barely see it on the graph. And you can see how all the technical replicates are really close. So these are CT values in qPCR. I'll admit this is not necessarily the first thing that people DOE when they start out, but to me, it just showed how in a single experiment, in one single qPCR plate, I was able to find a set of conditions that would now mean I could run qPCRs in a very precise and robust manner. So that was super exciting. And if I was already bought in theoretically before, I definitely saw the value as I saw these results. But let's not ignore the elephant in the room. DOE gets complex. It's a complex experiment to run, and there's a variety of challenges associated with it. And Mark has already alluded to a few. We can group them in 2 different types. We have practical challenges around the tooling that people use. So the traditional tooling that scientists are used to, mostly spreadsheets, doesn't really support DOE, and dedicated tools such as Design Expert or JUMP may require significant expertise. On top of that, the design of the experiment and the execution plan, what you're actually doing in the lab, are still disconnected. So often we're thinking of a DOE, we think about the concentrations of the liquids that we're using. Then we need to convert those onto volumes that we're actually pipetting ourselves or the robots. And there's the design aspect that can be challenging, but then there's the converting that into an executable plan that is also quite difficult. And then at the end of the experiment, we have a lot of data that we need to align back to the design and ideally include also any metadata that was generated during the execution. And doing this without this context can be both high touch and intensive, but also error prone. On the other hand, we also have adoption challenges. For most scientists, this is a new methodology, so it requires new mental models and requires time and expert input. And often that is a limiting factor. Scientists are busy, and learning a whole new way of doing experiments, especially if they've already been doing traditional experiments for a long time with good results, is challenging. It takes some time to become proficient in DOE, knowing when DOE should be used and how best to apply it. There are different strategies depending on what you're trying to do, from screening a whole range of different factors to finding a design space that is robust. And finally, when these requests come from top down, there's significant investment that is required in terms of training, scaling, and supporting DOE across entire organizations. And so this is one of the reasons why Synthace has always been invested in DOE, is trying to make life easier for scientists who want to use this methodology that we're all passionate about. So how does Synthace make DOE a reality? For those of you who don't know, our platform allows you to digitize an experiment from beginning to end and automatically creates instructions that can then be executed in the lab, either manually or by a dispenser or a liquid handler that we support. We've now brought in DOE to the heart of our platform. And so this is the sort of process you might go through, which is what I'll be demoing throughout this webinar. So first, you can create a design within the platform itself, or you can import a design that you've made elsewhere. Synthace can then automatically generate liquid handling instructions, so that disconnect between the design and the execution plan is completely handled by Synthace automatically. Finally, at the end of your experiments, we also can either automatically retrieve the data if it's been generated by an analytical device that we support, like the Claristar plate reader, for example, and we structure that back onto the design and onto any metadata that might have been generated, such as well locations, for example. And then now we also offer within the platform a way for you to analyze your DOE immediately, suggesting a happy path that takes you from your design to the model generation and validation. So let's contextualize this in an example before we go to a live demo. So let's look into an assay development example where we have an enzymatic assay that we're trying to optimize for. And so we're trying to increase the signal that we can measure from this assay. We have access to a dispenser. So we're going to be doing it in a 384 well plate. And some of the factors we'll consider are the type of buffer that's being used, the type of salt that is being used, but also we're going to vary the concentration of salts. And so in this case, what we want is that when one specific type of salt is used, the other is not. So mutual exclusion of the two factors. We're going to add a couple of additives such as reducing agents and BSA. And then we're also going to test if different temperatures can affect the outcome of this assay. So let's just go into the platform. So here I'm in the Synthace homepage and you can see a list of my experiments. I've already written some context to this experiment. And the goal, as I mentioned, is to test a variety of conditions and identify the ones that can optimize enzyme performance. And these are the factors I'll be looking at: buffer, HEPs or TRISH, salt, sodium chloride, potassium chloride, varying between 1 and 10 millimolar, BSA, testing 3 different levels of BSA. TTT has a reducing agent between 2.5 and 25 millimolar, and enzyme, and we're going to keep the substrates constant at 10 micromolar. And I already have a pilot workflow here. So a workflow is how we define a protocol in Synthace. Now, if you're not familiar with the Synthace platform, you can just create a workflow that suggests what is happening and dictates the steps for your experiment. And each of these blocks is responsible for a different set of actions. So this group of blocks here, we're defining the liquids that we're going to make available for this experiment. So here we have just water. And here we have a very special element that can automatically calculate what is the stock concentration that you should prepare in order to satisfy your DOE design. So here I've already specified the liquids we'll be needing. So your salts, your reducing agents, BSA, enzyme, substrate, etc. And I've specified also the maximum concentration at which they're available. And this could be because you've bought in a commercial kit, commercial reagent that already is prepared at a specific concentration, because you know the solubility limit of a specific salt, or simply because you can't be bothered to make new buffers and your lab mate next door already has some trish made up and you can just borrow some. So this is like your starting point. And Synthace will decide whether you can use these concentrations or you should use slightly more diluted stocks. You can also specify then the maximum number of stocks you wanted to make and the minimum transfer volume. Depending on the hardware that you're using, you might have an idea of just how accurate and how low they can go when it comes to pipetting. And so you can specify this volume here if you want to make sure that you're pipetting accurately. So then we just group all these liquids together and we feed it to the heart of this workflow, which is an element that just prepares mixtures. So in this case, I have one single mixture prepared, just as an example. So you can see that this mixture will have 0.1 BSA, 2.5 millimolar TTT, etc. The diluent we're using is water. We're making to a total of 100 microliters in a 384 well plate. You can choose what plate type you want to use, and you can see all the information about the plates and choose from our library. So I'm just going to simulate this at first, especially because some people might have not seen a demo before the Synthace platform. So when we click simulate, we get a simulation of all the steps of our experiment. So this page here, we have an overview of all the resources that are going to be needed. So we have our reagents here and their concentration. and the volumes that we'll need. We have a setup page that tells us how to set up our input plates in the first place. And we can also see what our assay plate is going to look like throughout the experiment. In this case, we're just making a single reaction. And we have the preview, which can show us in detail, step by step, what is going to happen. So first, we have 25 microliters being transferred, 20, etc. So this is good. This is what I want to happen in this experiment. But let's go back to the builder and actually make this DOE. We've now have, you might see here, this button saying DOE. Now, what this does is it changes the focus from building and specifying the steps that we need in our workflow to creating a design. When I click here, you'll see the entire canvas is grayed out, except for this element here, suggesting that this one is the one that can be viewable. When I click on it, you can see that instead of seeing all the parameters that we had before as options, you can now just see which parameters I can choose to set factors in my DOE. In this case, we can set the mixture compositions, which is obviously the main thing we'll be concerned about, but also, for example, the final target volume. If I wanted volume to be a factor in my DOE, I could set it here as well. So when I toggle this, I can click, I'm taken to this menu that allows me now to define my factors. And so if I click add, you can see that we can add 3 different types of factors, a new standard factor that can be continuous or discrete, a new mutual exclusion. So when we want to use, which can be used for categoricals, for example, but also for when we, and I'll show the case of the salt, where we have 2 different salt types, but we're also varying their concentration or constants. So let's start by defining a new factor. So when I click factor name, all the liquids I've already defined appear as suggested. So BSA, choose the unit, in this case, weight per volume, and set the values for my DOE. In this case, I'll set it as a discrete factor. And now we've specified the first factor in our DOE. I'll add the mutual exclusion factor now. I'll call it salt. I'll want to test both salts at the same levels. So I'll toggle this so I don't need to specify it twice. And now here, I'll just find my potassium chloride and set this to be 1 and 10 millimolar. And my factor. And now my sodium chloride. This is already defined, factor and mutual exclusion. So now you can see we have this factor. And so whenever potassium chloride is used, sodium chloride is not, and vice versa. Our buffer is also categorical. In this case, we're only going to use one concentration. And so it's either happy 20 millimolar or Trish. Great. And now I've mentioned we're going to add some additives. So let's add the reducing agents. And that was between 2.5 and 25 millimolar. Now, in this case, I'm going to set the sampling to range. And that's so I can show you how a Spacefil design works. So in this case, instead of just sampling between either 2.5 or 25, it can sample a whole range between 2.5 and 25. So it could sample 3 or 5.6. But what I don't want, I also want to include zeros. So I want a few runs that don't have any DT. So I can include this here. But what this means is that 3 runs are going to have no DTT, and then all the others are going to range between 2.5 and 25. So this allows me to exclude this very small range between 0 and 2.5, where maybe the effect would be very similar to a 0, but it would be incredibly hard to pipette by a robot. Okay, so this is an enzymatic assay. We're still missing the enzyme. The BSA is already there, so let's add the enzyme. You can see we support a variety of different units. And so in this case, I'll also sample a range. I will not include any zeros because I'll specifically include a control that has no enzyme just to see it has a negative control. And finally, we need a constant. We need substrate to feed our reaction. So I'll just add this as a constant. 10 micromolar. Great. As I mentioned, we also want to check temperature. Now, temperature is not a factor that will affect my liquid handling. So it's not associated with this specific element, but it's a custom factor. So I'll just click new factor, temperature. This is numerical. And I'll test, say, 28, 32, and 37. Great. So I think this is our design setup. So all our factors. Now, if you want, you can export these factors, and we can export them as a JSL format as well. So if you want to build a specific design in Jump or in any other tool that you might use, you can, and then you can import that design back to Synthace, or you can continue on this journey within Synthace and go onto Calculate Design tab. Now, here you can see that we'll be able to offer a variety of different types of design. You can have a space fill design, which is the one where I mentioned we sample throughout an entire range across levels, optimal designs, full factorial designs, or you can upload your own. We have some information here also explaining how each one of these works. So below you have our diagnostics tabs. So this gives you an idea of the power of the current design and the distribution of each data point throughout your design. You can see your correlation matrix, and you can also model effect size. So if we know actually this, I've already done some pilot studies, and I know that actually the signal tends to be really good. I know I'll expect an effect that's a bit higher. You can model it and you can see how it changes. The design is also here. You can see, for example, if we look at between this and Trish, that when one is defined, the other is set to 0, similarly to our salts. So you have here 0 potassium chloride when we have some sodium chloride. But we can add some replicates as well. So this, we only have 96 runs. As I mentioned, we are doing a 96 384 well plate. So let's add some replication. We can choose if we want to keep all the replicates together, repeat the run order. So we have one set of runs and then the other, or fully randomize the design. And then as we do, the diagnostics gets updated, and you'll also see here that we now have 384 runs. But let's add some controls. So for that, actually, I want to control. So I will reduce number of runs to make sure I still just fit everything in one plate. And I'm going to add a no enzyme control. So I'll set enzyme here to 0. And then I'll add also a no substrate control, which I'll set to 0. Cool. Oh, there we go. I'll save these. And so now you see that the controls will also be replicated 4 times. And so we have 384 runs, including 8 controls. Right now they're appended to the end. So you can see them here at the end of the design, our 2 controls. But we can also randomize it or get a control repeated every certain number of runs. So actually, if you're simulating it as a device and your design is spanning multiple plates and your device supports having multiple output plates, which is common in the liquid handler, but less so in the dispenser, you can use this to make sure you have a specific control in the same place in every plate. Okay, so if we're happy with our design, now we can just simulate it. And now you'll notice that the simulate button no longer is set to simulate on its own, it's simulating with a design. And so what this is going to do is load the DOE design on top of the protocol we had already defined. And so you'll notice here a difference between it's tagged as part 1, whereas this just says view simulation. And this is letting us know that this is a specific DOE design. And I'll show you an implication in a minute. But let's look at our simulation now. So again, we have our resources overview page. You can see now that we need more liquids. You also see that we need different stocks. So we have 2 entries for BSA. One is at 2 grams per liter. One is at 20 grams per liter. And that's because Synthace automatically identified that to satisfy my design, we need to have 2 different stocks of BSA, because if we use the high concentrated stock, we might end up pipetting a very small volume in some runs. And if we use the slightly more diluted stock, maybe we would go over the total volume that is allowed. We can see our design here as well, and we can download it to make sure alongside with temperature as well. And if we go now to our preview page, we can see that now we have an execution plan that creates 384 runs. And so very quickly, we went from specifying the factors and the levels that we need in our experiment to actually creating a set of instructions that device can use. If I click on any given, you can see all the liquids that have gone into that. You can see that this refers to run 107 and the set points for each one. And I can now just click download instructions, and this will give me the file that I need to go and run this on my Dragonfly, which is the device that I simulated this against. Now, if you've been paying attention, you might wonder, you specify temperature has a factor. How are you gonna test the different runs that are all in the same plate at different temperatures? And that's a really good point. I clearly made a mistake. It's a good thing that I'm doing this all in silico and not actually pipetting just yet. I can check for any problems before I actually encounter them. So if we go back to our design, one thing that I can do is come here and group the runs by a specific factor. So I can group the runs, in this case, by temperature. And as you can see now, this results in 3 simulations. That means that now we're creating 3 simulations, each one for a specific temperature. So if I go back to simulate with design, what you'll find now that instead of 1, we're creating 3 simulations at the same time. Each one will follow a specific part of the design. Temperature set at 28, temperature set at 32, temperatures set at 37. Try saying that 3 times fast. So if I open 2 of them to compare, if I go to the design tab, you'll see that now all these runs are set at 28. And in part 2, all of them are set at 32. But Synthace is still aware that this is only a subset of the design. So I can go on the full design and you can see all the runs and highlighted are the ones only associated with this execution. And in fact, if I go to my preview all the way to the end, I can see that now I only have a subset of the runs. And on the other simulations, I have all the other runs. So now I can run this 3 different times, get each plate to its own incubator set at different temperatures, and then analyze all the data together. And so you might wonder, okay, that's neat, but now I need to keep track of all the 3 different simulations. This is something that can become a bit harder to do. And so while we've been playing and simulating, everything has been tracked in our experiment map. So if we go back to our experiment and we click here, the experiment map now shows us this is the workflow we started with. This is the one we simulated against the 3 different temperatures that created the 3 different simulations. So part 1, 2, and 3. So everything has been logged automatically for you. And this will become in handy as well when it comes to data analysis. So let's imagine then that we have gone to the lab, we've created a design that we want to generate with all the liquid handling instructions, and we've gotten some data that we've already uploaded to the platform. So now we need to collate and structure these DOE results and do our analysis. But I would just like to highlight that, we very quickly, in a matter of minutes, went from building, designing, creating a design, specifying the factors and the levels. And admittedly, that can take quite some time just doing the research to identify the right set points. But then converting that design, defining that design, and converting it to an execution plan was something that we were able to do within the Synthace platform in a matter of minutes. So let's imagine we ran this experiment and we had 3 different executions, all with the data for the 3 different temperatures. So we ran this experiment. We actually ran it in the lab with 3 different temperatures. And now what you can see in the map in yellow are the executions. So these have been marked as complete and have data associated with it. If I navigate to one of these, you can see in the results that we have data uploaded. And so I can just click structured data. Now, immediately Synthace is aware that this execution is part of the series. So he highlights that before I'm about to structure data, that there are two other executions that should probably be considered because they're part of the same DOE design. And so this is what this pop-up shows, is that we have two other executions part of this 3 execution design. They all have data associated with it. And so I can choose to select the other 2 and structure all of it together. So now what you can see here is we have our design, but now we also have this signal column. The signal is the data that I measured, that I captured in the lab. And you can see that all the executions were automatically structured. We have here set points of temperature at 37, 28, and 32 degrees. So all of this was automatically captured and structured using Synthace. Once we're happy with it, I can include some extra columns of metadata from the experiments, and then I can click Start Analysis. That immediately goes to the analysis page. The analysis page is something I'm going to cover in a minute as well, but you can see that what has happened in the background. So I'll just go back a little bit. When I click to go back to the experiments, what has happened is that now Synthace has mapped automatically that these 3 executions are linked together to a data node, and that data node is then taken onto an analyze data node. And all these are links to the different apps that exist within the Synthace platform. So what can we do when it comes to analysis? I hope it's become clear as well that at any given point, I could download the data, the structured data, which already has given me a lot of value, and upload it to a different platform and use their analysis tools. But we want to bring everything we want to make the process as streamlined as possible. So we also offer some analysis tools. They're comprehensive enough for most DOE applications, but at any given point, you can continue elsewhere. So the first thing in the analysis page is what we call the quick view responses tab. To me, this is quite important. I think as a scientist, we're all familiar with the idea that an experiment is running and we're just hovering over the plate reader, trying to see if we can have an idea if the experiment has worked or not. Maybe there's a visual clue on the plate itself or some easy plotting that we can make so that we immediately attenuate the anxiety of running experiments. Running experiments is really hard. People get, and understandably, slightly emotionally involved. And so you just want to go home at the end of the day knowing that, yeah, it probably worked. I'll look at the data more carefully tomorrow. And so this view, it gives you just that. So immediately we're plotting our response over a variety of different factors. So here we have our BSA, DTT, enzyme, and temperature. So immediately just looking at this, it means that there seems to be an effect here with temperature where the optimal seems to be at 32 degrees. And so this is likely a quadratic effect. Enzyme, unsurprisingly, seems to have a positive effect. More enzyme means higher signal. TTT potentially also has a sort of quadratic effect, which is not necessarily that unexpected in biology where everything happens within Goldilocks conditions. So not too much, not too little. And so shapes like these can happen. And with BSA, it's hard to know, but perhaps there's a slight negative effect. What you can do as well is immediately decide to create plots, other things. We have many other factors in our DOE. And so we can just come here and, for example, create a new plot that looks at the effect of salt in our response. And in this case, because we had 2 different types of salt, we can plot them in different colors. Let's try to have an idea. So it doesn't seem to be very clear. Perhaps one is better than the other. So we can just plot the 2 together and see, but actually maybe looking at this again, the two plots seem pretty similar. And so perhaps actually this is a good finding in itself, because it means you're no longer dependent on supply chain for one specific chemical. You can easily swap between one or the other if you want to. This is again just a quick view of the data. We can also view it on a plates layout form. And so if you had any specific edge effects like evaporation, these would become obvious here, and you could now exclude those runs from your analysis because they would be artifacts. Which takes me to the next stage, which is the select and transform data. So here you can see how signal over run number. So if there's any specific effects simply due to run number, we know that we have a problem with our data sets. You can also see if the results are normally distributed or not. And you can see that both in the histogram and normal plot. And if you have any issues here, that would suggest that you might need to apply a transformation. Again, you might need to apply a transformation. We offer the standards. We can apply a log transformation, box-cox, or you can write your own Python expression if you know the best way to transform your data or some sort of data transformation you need to do to actually get the response that you care about. And then we just need to find the effects. So in this case, Synthace has already automatically pre-selected all the effects that were below 0.05 p-value. And you can see how the model would look like immediately, what's the prediction for each one of them. Now we know that actually that's maybe we want to be a bit more conservative. We can manually exclude any terms that we don't want to have in the model. So in this case, I've excluded everything that wasn't smaller than 0.01, and now we can model those terms. And so what you see here is, again, all the terms plotted over each other. So in green, we have our main effects, and then all the interactions are mapped in purple. So for example, here we have the interaction between temperature and BSA that is in dark purple, so it's selected. And then here we have the quadratic term of temperature in orange. Immediately on the side, you can see the R-squares for this model, the residual standard error and the p-values. And you can also see here below the coefficients, estimates, and the error associated with each one. That gets automatically recalculated as you switch the terms that you want to include. You can create a variety of models, save them. In this case, this one was called manual because the terms were selected manually, but we also offer different types of regression techniques, specifically stepwise regression and lasso regression. You can see your predicted over actual plots, residual over predicted, and normal plot of residuals to make sure that your model is sound. But especially if you're perhaps less confident at first on what makes a good model, you can actually create different models and use the automated regression techniques and just see how similar they are. I would like to believe that if an effect is significant enough, if it's real, then a variety of models should be able to pick that up. And so that could be a way that you could easily compare different models and see what type of results you get. And finally, we can also then explore the different models. I think here I'm creating a model that only has main effects. You can see that the R-squared value are immediately much lower because we saw the shape of temperature, for example, and we know that can't be a good model. And so when we explore model, you get to see your prediction profiler. So what you have here is the different factors and how the model predicts how they affect your signal. And we can maximize or minimize the response to find the optimal values for your response. So here, for example, we're plotting BSA over salt, but we know that temperature, as you can see, has this curvature associated with it, and so does DTT. So this allows us to very quickly see this effect very clearly. And actually, if we plot one heat over temperature, you see a nice kind of dome that suggests that, yeah, here is your maximum. And you can easily fall off a cliff if you deviate too much, either too high or too low DTT or too high or too low temperature. You can maximize, find the biggest value. So it sounds like we don't need BSA at all. DTT is here in the middle, temperature as well. Salts, there doesn't really seem to be a difference, and the highest amount of enzyme, the best. Sometimes we may also have multiple responses. So for example, in media optimization, you might be checking for the production of a specific chemical, but that you want to increase, that you want to maximize, but you know that there's maybe a side product that you want to minimize. You could actually include both responses at the same time, create different models for each one, and maximize one and minimize the other. which you could do here. And finally, just as a way to give you some sort of easy way to compare a variety of different models against different responses, we have this traffic light system where for each model here, you can see different colors representing the R squared, the p-value, and the normality of residuals. And so you can see that, for example, the main effects only model is clearly not very good because the residuals are not normal and the R square is pretty bad, but all the other models are actually pretty good. And so again, we hope that this kind of gives someone, especially if they're a bit more new to DOE, a sort of streamline of what the process of analyzing our data looks like. And at the same time, provides people who are familiar with DOE, who want to look at specific types of plots, the option to do. And so if I can just summarize what the new DOE product in Synthace offers, is that we can convert DOE design to an executable plan seamlessly. We can link design and generate the data automatically. We can connect and align experiments in the same campaign. And we can analyze your data, everything all in the same platform.
Thanks very much, Nuno. It's hopefully a nice explanation of, it was a nice explanation of the platform. Hopefully everybody can get a good idea of what it can do. So just to kick off, obviously this webinar has intentionally been focused around the Synthace platform and how it can help do DOEs. But we've covered this in previous webinars, but just for anybody who's new, how might they maybe get started with DOE just to get going in the absence of Synthace in the first instance?
Yeah, that's a really good point. I definitely remember going on that journey. I think here at Synthace, I was immediately thrown DOE Simplified Copy, which is a great book to get started with DOE. From there, you can move on to a variety of different other content. I think we also have Sid has once written a blog post with a variety of different books that he recommends, and he's a DOE expert. Then the other thing that I would suggest is that I think because it can feel a bit overwhelming at first, and experiments can be costly, taxing, so people want to make sure they get them right. So then that almost stops you from doing DOE because you want to make sure that it's perfect, and you just don't have the confidence because you've never done it before. So I think a really good advice is to just do it. Your design doesn't need to be perfect. Once you generate your data, you can also analyze it in different ways and you can correct it. But just going through the process, understanding what it takes, and also seeing the value that you'll get from it immediately should hopefully give you that confident boost. That means, okay, cool, I will now invest time in learning this properly. It makes sense because however long I spend now, the next 3-4 weeks learning about DOE and not going to the lab and do bench work will save me months down the line because I'll be doing better experiments going forward.
If there was ever one kind of objective of us putting these webinars out there, then it's very much just to get people over the hump of doing their first DOE, because in my opinion, that's the point at which people get hooked and realize how powerful it can be. So we've had a few more specific questions coming in. One here from Catherine. Hi, I may have missed this comment, but how are the DOEs generated? Is it an optimal design? And if so, what is the optimality criterion? Thank you.
Yeah, we offer 4 different types. So we offer a space fill design, where you're testing the entire range between the levels you specified. We offer an optimal design, full factorial design, if you want to go down that road, or you can upload a design that you've generated elsewhere. And so I believe our optimal designs are like standard the optimal designs. I can look more carefully into the details.
So yeah, in a little bit more detail, because I actually asked one of the guys at the heart of this during the webinar when this question came in. So yes, for any screening design, it will be de-optimal. But as soon as you're trying to model quadratic designs, which might suggest that you're actually optimizing at that point, then it will go for I-optimal instead. I won't go into the details of what those terms mean so that we don't go into the really deep stats, but hopefully that's helpful for the more statistically inclined. And then a follow-up question from the same person. In this example, is temperature a hard to change factor?
It wasn't, but you can specify it. So for all factors that you specify, you can tick the box for hard to change, which should add extra replication of that factor because it's taking into account the fact that you will be grouping runs that have this factor together, and you run the risk of aliasing with other factors in your design. So you want to balance for that. So that is something that we've built into the platform. Any factor can be marked as hard to change factor.
Yeah. So in this case, we were actually making the assumption that splitting it across different plates wouldn't have a dramatic effect. And often we found this when we're just doing pragmatic designs within biology, is that we will sometimes consciously make an assumption about how a particular setup or a particular design might have an impact on the noise or the variance across a design. For example, in the past, we never used to randomize our run order because running these by hand, it was an awful lot easier to keep it in standard order to run them. And we did so fully cognizant of the fact that could give us some systematic bias if there was some kind of effect across the plate or whatever. So these are the kind of things that people can think about when they're designing. But yeah, if you are worried about that, then yeah, if you stick on a hard to change factor, then Synthace will automatically put in the right kind of blocking methodology to make sure that you don't have any systematic error.
On a related note, the other thing that you can also create is what we call quasi-replicates. So you can replicate an entire design based on the level of the factor. In the context of this specific case, if you're concerned that any liquid in your mixture could affect the background signal, you can duplicate the entire design with and without enzyme. And so you could have half a plate with enzyme, half a plate without enzyme. All the runs match exactly except for the enzyme term. And so you can calculate one by one. And actually, you can use this as some sort of background calculation that could vary. If it could vary between runs, then you can normalize specifically.
Okay, I've got two more questions I'd like to get through. So can your control of the automation employ multi-dispensing?
Yes, if your device supports multi-dispensing, we can set the liquid classes on the build mode to use multi-dispense.
And then from the results shown in which the largest amount of enzyme was preferable, would the DOE be expanded to include higher levels? So I guess this is talking about then iterating from these initial results.
Exactly. So then as part of a DOE campaign, you might have a strategy where you iterate depending on your goal. And so then, yeah, you could push these levels forward until you find a value where perhaps the signal saturates, right? And you could still, and there's, from that moment onwards, there would make no sense to adding any more enzyme to it. But yes, that's quite a common strategy, right? You screen a variety of factors, and then you might iterate to try and push the levels outwards a bit more to the area that is giving you the optimal response.
Yeah. And I guess the whole point of what we've done with the Synthace platform is we've just tried to make it as easy as possible to run those iterations so that then you can move rapidly through that design space depending on what the results show you. Okay. So with that, we'll conclude the Q&A session. So the DOE Masterclass webinar, if you'd like a demo, then it's all on the website. Thank you very much to our audience for participating. And we hope you enjoyed today's talk, and we look forward to seeing you on our next webinar.
Thanks so much, everyone.
Thank you very much.